many carpels are there. All this is called as morphology. But anatomy represents what are the internal structures or what is the internal construction of these plants that comes under anatomy. Are you able to understand? So how can you define anatomy? Anatomy is the study of internal organization. Okay, ma? Or the study of internal parts is called as anatomy. Okay. So that's what he gave here. Anatomy is the study of internal structure of plants and other organisms. Clear, Nama? So first of all, we will study about tissues. So how many types of tissues are present? So and what are their functions also? That first of all we will study. Clear? So what is meant by tissue? Basically, tissue means what? Group of cells which perform a certain function is called as tissue. Are you able to understand? So it is a group of cells having common origin and function. So their origin is common and their functions are similar. So this group of cells we can call them as tissues. So based on the capability of a cell division, plant tissues are of two types. Clear, Nama? So based on how cells can divide, we are dividing the total tissues or cells into two types. So what are they? First one is called as meristematic tissue and the second one is called as permanent tissue. Are you able to understand? So based on capability of cell division, it means how cells they are able to divide. See, these meristematic cells, they have the ability to divide continuously. Are you able to understand? Whereas permanent tissues, already they are matured and they doesn't have the ability to divide. So based on cell division, we are dividing the tissues into two types. One is meristematic and the other one is permanent tissues. Are you able to understand? If cells are dividing continuously, then we call them as meristematic. And if the cells, if they have already matured and stopped further division, then such cells we can call them as permanent tissues or permanent cells. Understood the difference? No? Okay. So next we are studying about meristematic tissues, which is also called as meristems. Clear, Nama? So these are the tissues where active cell division and growth occurs. It means continuously cells are divided and growth happens. So these regions you can call them as meristematic regions and these tissues you can call them as meristematic tissues. So if a cell division is happening continuously, then the type of tissues you can call them as meristematic tissues. And based on the position where these divisions are taking place, again we are dividing these meristematic tissues into three types. Are you able to understand? First one is apical meristematic tissues, second one is intercalary meristematic tissues, and the third one is secondary meristems, or you can call them as lateral meristems also. Are you able to understand? So these two, apical and intercalary, you can call them as primary tissues. Are you able to understand? And why they are called as primary tissues? Because these are the tissues which are formed at the starting of the plant life. When plant starts growing, then first of all you will see these two tissues. Which tissues, ma? Apical meristems and intercalary meristems. మీ ఎన్సీఆర్టీ బుక్స్ ఎక్కడ? ఎక్కడ మీవి? ఎన్సీఆర్టీ బుక్కులు లేకుండా క్లాసులు రాకూడదు. మీరు బుక్స్ తీసుకోండి ఫస్ట్. క్లియర్ మా? సో apical meristems and intercalary meristems, you can call them as primary tissues, and the secondary tissue is called lateral tissue or lateral meristem. Okay, ma? So already we have understood why they are called as primary tissues, no, ma? Because they were formed at the initial stages of the plant life. That is why they are called as primary tissues. Clear, no? So here in the figure you can see apical meristems. Apical means what, ma? They are present at the topmost positions, either at the top of the plant or at the bottom of the root. Clear? They are called as apical meristems. And intercalary meristems, they are present in between two nodes, and lateral meristems, these are the secondary meristems. Clear, no? Now let us study each of them in detail. First of all, we will study about apical meristems. Clear? So total meristems we have divided into how many types? Three types. First one is called as apical meristem. So they occur at the tips of roots and shoots, and they produce primary tissues. Are you able to understand, ma? So what is meant by apical meristems, ma? Where they are present? Apical meristems, they are present at the bottom of the roots and the top of the plants. Okay, so that is why they are given two names, ma? Root apical meristem. Root apical meristem means you will find them in the roots. Shoot apical meristems means you will find at the topmost regions of the plants. Clear? So root apical meristem and a shoot apical meristem. Okay. And here there are some structures which are given. Once let us see them. Clear? So this region here, you can call it as the root apical division, or this is called as the root cap. Clear, Nama? So for the bottommost part of the root, there will be a cap also for protection. So roots will be very tender, Nama. For protecting these roots, you will have a root cap. Then this innermost region, this region you call it as central cylinder. Clear, no? And surrounding this central cylinder, there will be one more region which is called as cortex. Clear, no? So central cylinder is surrounded by what? Central cylinder is surrounded by cortex. Clear, no? And in region where central cylinder and cortex are present, here you will find the root apical tissues, or those tissues which divides continuously. Are you able to understand? So here you are seeing this region, no? They are called as root apical meristems. Okay. And surrounding the cortex, again we have another layer which is called as protoderm. What is the second another layer which we have? Protoderm is the another layer. Clear? So then you can see the initials of central cylinder and the cortex. It means the starting positions of central cylinder and the cortex you can see. And you can see the initials of the root cap, and that is followed by the root cap. Are you able to understand? So where you are finding this root apical meristems, what is the position of root apical meristems? You can find them in between the central cylinder and the cortex. Are you able to understand? So the middle part of the root is called as central cylinder. Central cylinder is surrounded by what? It is surrounded by cortex. And these root apical meristems you will find in the gap between this cortex and the central cylinder. Are you able to understand? So this is one point which you should remember very carefully. So therefore, what shall we write here? Root apical meristems. Root apical meristems. Meristems means what? Cells, that's all. Cells which divide continuously. Clear, ma? Where they are present, ma? Present between central cylinder and cortex. Okay. What is meant by cortex? Cortex means the outer layer which is surrounding this central cylinder is called as cortex. So in the gap between these two, you will find the root apical meristems. Fine, ma? So these are cells which divide continuously. Similarly, if I go to the shoot apical meristems, clear, ma? So here you have a leaf primordium. What is meant by leaf primordium, ma? So this is the region from where leaves will originate. Able to understand, ma? Regions of leaf origination. They are called as leaf primordium. Able to understand? And below this leaf primordium, you can find the shoot apical meristem region, or this is called as shoot apical meristems. Are you able to understand, ma? Then some of the cells of shoot apical meristems, they are present here and they are called as axillary buds. Okay. So in axillary buds also, what kind of meristems you will see? You will see the shoot apical meristems only. Are you able to understand? And then we have a differentiating vascular tissue. Are you able to understand, ma? So therefore, these axillary buds, we will write one important point about axillary buds, ma. So here also we find some shoot apical cells. So these cells also they will divide, no, ma? And what is meant by bud here? Bud means what will develop? From the nodes, leaves will develop, no? From the buds, what will develop? Flowers can develop or branches can develop. Are you able to understand, ma? So for the branches and flowers to develop, definitely cells should divide, no? And here also what kind of cells you will find? Here also you will find shoot apical cells only. Are you able to understand, no? So these are the positions of the apical meristems. So apical meristems you are dividing into how many types, ma? One is a root apical meristem and a shoot apical meristem. And what are the positions also we have explained them. So then you should understand and by heart the positions of these cells. Clear? Next discussing about root apical meristem. So it occupies the tip of the root. So where you can find this root apical meristems, already we have studied now. They are present in between where? They are in between the central cylinder and the cortex. So that is nothing but the tip of the root itself. Fine. And still anything we have studied, already we have studied all of them. So see here, clear ma? So this is the central cylinder, this is the cortex, and surrounding the cortex we have one more layer which is called as protoderm. Clear nama? And this structure you can call it as the root cap. And here you will find the root apical meristems. Clear nama? So the region, this region, here you will find the root apical meristems where a division of cells will take place continuously. Clear? That's it. So go through the diagrams very carefully, then you can understand very easily. Then the second type of apical meristems is shoot apical meristems. And where they are present already we have studied them, they are present below the leaf primordium. And some of the shoot apical meristems they are present at the axillary buds also. Are you able to understand? So you are seeing this region Noma. So here also you will find shoot apical meristems. Here also you will find some shoot apical meristems. Are you able to understand? Okay. Now let us study what he has given here. So it occupies the distant most region of the stem axis. What is meant by distant most region of the stem axis? Topmost part of the stem. Topmost part of the stem is nothing but distant most region. So some cells left behind from the shoot apical meristem constitute the axillary bud. That's what we have studied now. Some cells they are left behind from the shoot apical meristems. అంటే కొన్ని cells వెళ్ళిపోతాయి అన్నమాట. So they are left behind. So these cells where they go, they are present at the axillary buds. And what is the function of axillary buds? These axillary buds only they develop into flowers or they develop into branches. Are you able to understand? So here also we have one group of shoot apical meristem, and here also we have a group of shoot apical meristems. So in two positions you can find the shoot apical meristems. Are you able to understand? That's it. So they are present in the axils of the leaves and can form a branch or a flower. Where you can find buds? Buds are present at the starting of the petiole, no? Buds are present at the starting of the petiole. What is a petiole? Supporting structure to the leaf is called as petiole. For example, if this is a stem, so consider this is a leaf. Are you able to understand? This structure, what we call, this structure is called as petiole. Okay, ma? And at the starting of the petiole, you can see these buds. So from these buds only, you can find branches or flowers to be coming out. Okay. So at the axils of this flowers, leaves also, which are called as buds, you can find a shoot apical meristems. So this is the position of shoot apical meristems. Clear, Nama? Okay. So then we study about the second type of meristems, which is called as intercalary meristems. Are you able to understand, ma? So already we studied that meristems are of three types, no? One is apical. Apical is again divided into root apical, shoot apical. Then we are coming to the second one, which is called as intercalary meristems. Now let us see. So they occur between mature tissues. They occur between the mature tissues. What is meant by mature tissues, ma? Mature tissues means what? Permanent tissues. It means these cells, they cannot divide further. Able to understand, ma? In between these two permanent tissues or mature tissues, you will find intercalary meristems. Are you able to understand, ma? And they occur in grasses and regenerate parts removed by the grazing herbivores. Clear, ma? So these intercalary meristems, where you will find them in grasses also. And browsing animals or grazing animals, they come and they eat the grass, no? They cut the grass. So to again regenerate that cut grass, these intercalary meristems are responsible. Are you able to understand? Okay. And one more important point here we have to study is this one. So intercalary meristems, this is the structure in monocot stems only. Are you able to understand, ma? So here you are seeing this brown color region, no? Clear, no? What is this brown color region? That is the region where intercalary meristems are present. Are you able to understand? For example, have you seen sugarcane? Sugarcane, there are nodes untai kada. If you cut them, then this is the brown color structures which you see. Clear? And these brown color structures are the regions where intercalary meristems are present. Okay. So then what is this region? This region is what? This region is the region of mature tissues. So here growth doesn't happen. Are you able to understand? Only at the nodes only or the separations only you can find intercalary meristems. Are you able to understand, ma? So this region I can call it as intercalary meristem. This region I can call it as intercalary meristem. And this region in between, this is called as mature region. where you will find the permanent cells or permanent tissues. Clear now? And growth happens in which direction? Growth happens in upwards direction. Okay, ma? It means the cells, they will continuously divide at the intercalary region and the stem starts growing in upwards direction. Are you able to understand? So what is the position of intercalary meristems, ma? They are present in between the permanent tissues. First you will find permanent tissues, then intercalary tissues. Then you will find permanent tissues, then you will find the intercalary tissues. So this is the arrangement of intercalary meristems. Understood now? Okay. Then some more definition he gave here about intercalary meristems. Clear, ma? So apical and intercalary meristems, they are called as primary meristems. Already we have studied, no? Apical meristems and intercalary meristems, what you call them, you can call them as primary meristems because they appear early in the plant. Because when a plant starts growing at the early stages, then you will find these meristems, apical and intercalary. That is why they are called as primary meristems. Clear, no? So, and are the early plant life and contribute to the formation of primary plant body. So, during that, specific regions of the apical meristems produce dermal tissues, ground tissues, and vascular tissues. So, this is one important point which can be asked in the examination. Are you able to understand, ma? So, in these specific regions of the apical meristems produces what, ma? They produce dermal tissues also. They will produce ground tissues also, and they will produce vascular tissues also. Are you able to understand, ma? So, what is meant by dermal tissues, ma? Outer layers of the plant, they are present, you know. For example, outer bark of the plant, outer cells of the plant. So they are produced by whom? They are produced by this apical as well as intercalary meristems. They are called as dermal tissues. Outer covering to the plant is called as dermal tissue. Dermal means skin, no? Okay. Clear, no? Then ground tissues. First of all, we will discuss about vascular tissues. What is meant by vascular tissues, no? Vascular tissues means the tissues which are responsible for transportation. They are called as vascular tissues. Are you able to understand? And what is meant by ground tissues? The tissues which are not dermal and which are not vascular. The remaining all the cells you can call them as dermal, what you call ground tissues. Are you able to understand, ma? So neither dermal nor vascular. They are called as ground tissues. So are you able to understand what are the tissues produced by apical meristem and intercalary meristems? They will produce dermal tissues, vascular tissues, and the remaining tissues which are called as ground tissues. These three tissues are produced by the primary meristems. Clear, number? Then we are studying about the secondary meristems, which is also called as lateral meristems. Clear, ma? So the meristems that occur in mature regions of roots and shoots. Already we have studied. They are cylindrical in shape. Clear, ma? They are seen in gymnosperms and dicots. So this is another important point which you should remember. Are you able to understand, ma? So secondary meristems, where you find? Secondary meristems you will find in gymnosperms and dicot plants. Are you able to understand? And remaining in the remaining plants, you will not find this secondary meristems. Okay, ma'am. So secondary meristems, they are a combination of what? They are a combination of fascicular vascular cambium, interfascicular cambium, and cork cambium. Okay, ma'am. So all these three combined together, they are called as secondary meristems or lateral meristems. Are you able to understand, ma'am? So in secondary meristems, again, what are the group of tissues which are present, ma'am? So fascicular vascular cambium, interfascicular cambium, and cork cambium. What is the meaning of them? Again, we will study in the upcoming slides. So these are responsible for producing secondary tissues. Already we have studied. Are you able to understand, ma? So this is the position of the lateral tissues. Are you able to understand, ma? Okay. Then permanent tissues. Permanent tissues are nothing but lateral tissues. Are you able to understand? We are studying about them. The cells produced by primary and secondary meristems become structurally and functionally specialized and lose the ability to divide. They are called as permanent mature cells and constitute the permanent tissues. And they are of two types. So simple as well as complex. Are you able to understand, ma? So what is meant by mature tissues or permanent tissues in this condition? What is the meaning of this? So the cells which are produced in primary tissues. What is meant by primary tissues? Root apical, shoot apical, and intercalary. They are called as primary tissues. Nama. So these tissues, they mature. They mature and they will have their own function. Are you able to understand, ma? So it means what they are functionally specialized. Functionally specialized means each cell has a different function. Are you able to understand, ma? So after maturation, they lose the ability to divide. Further, they cannot divide. And once they cannot divide, they are called as what? They are called as permanent tissues, or you can call them as mature tissues. Clear, no? So, and again, these permanent tissues we are dividing into how many types? We are dividing into simple tissues and complex tissues, or you can call them as simple permanent tissues or complex permanent tissues. Are you able to understand? Na? So, ఈ permanent tissues ఎట్లా form అయినాయి చెప్పండి. These permanent tissues, they are formed from the primary tissues itself. Primary tissues, when they mature and when they get functionally specialized, further they cannot divide. And such cells we can call them as permanent tissues. That's it. Clear? So then permanent tissues again we have divided into two types. Permanent tissues we have divided into simple permanent tissues, complex permanent tissues. So simple permanent tissues. So parenchyma, Colonic, and Sclerenchyma. These are the examples of simple permanent tissues. Are you able to understand? So what is the definition he has given here? Tissues having all cells similar in structure and function. Okay? They are called as simple tissues. What is the meaning of this one? It means all the cells which are present in this parenchyma, they perform the same function. Are you able to understand? All the cells they perform same function. Similarly, all the cells which are present in colonchima, they also perform the same function. All the cells which are present in sclerenchyma, they also perform the same function. If every cell perform the same function, then the type of cells we can call them as simple permanent tissues. And a simple permanent tissues are how many types? Parenchyma, colonchima, and sclerenchyma. Are you able to understand the difference? So in simple tissues, all the cells will be similar and they perform the same function. But that is not the case in the case of complex permanent tissues. Complex permanent tissues, they are a group of different cells and they perform different functions. That is why they are called as complex. But they are simple because all the cells they are of same type. Clear, Nama? So then see about parenchyma. Clear? So what is the function of parenchyma? It forms the major component within the organs. Clear, na? So it means in the total organs of these plants, which are the major tissues which are present, parenchyma are the major tissues. It means they are majority in number. Okay. So cells are generally isodiametric. So all the cells of this parenchyma, they are isodiametric. Isodiametric means what? All of them almost they have the same diameter. That is called as isodiameter. It means the sizes of these cells are almost similar. That's what he's trying to say. But they may be spherical, they may be oval, they may be round, they may be polygonal, and they may be elongated also. So these shapes may be present, but diameter wise, they remain the same. Clear, ma? So walls are thin and made up of cellulose. So what is the nature of the material of this cells is made up of? This is also very important. Clear? So what about the walls of this parenchyma cells? Walls are thin walls are present and they are made up of what? They are made up of cellulose. So cells are closely packed or have small intercellular spaces. So in parenchyma cells, in between the cells, you will find some intercellular spaces. Some spaces are present. Okay. But if you go to other cells, you will find some cells where you don't find any gaps. But here in parenchyma cells, definitely you will find some gaps which are present. So either they may be closely packed or you can find some gaps also. Clear? And what are the functions of this parenchyma cells? They are responsible for photosynthesis, they are responsible for storage, and they are responsible for secretion also. So these functions also you should remember. Are you able to understand? So parenchyma cells, they are isodiametric and they are the major cells present in the plant body, and walls are very thin, made up of cellulose. You can find small intercellular spaces and they perform the most important functions of the cell, functions of the plant, which is called as photosynthesis, storage of food material, and the secretion of different plant hormones. Understood, Naman? That's it. So here I have already written something. The cells perform important functions. So simple permanent ground tissues that form the bulk of the plant tissues, such as soft part of the leaves, fruit pulp, etc. So these are the regions where you will find this parenchyma cells. Are you able to understand? So where you find them in bulk of the plant tissues. Bulk of the plant tissues means in most regions of the plant tissues you will find parenchyma cells only. Similarly, in the soft part of leaves also you will find parenchyma cells, and fruit pulp also you will find the parenchyma cells. Clear, Nama? Next, coming to colonchima. Are you able to understand, ma? So it occurs in layers below the epidermis in dicot plants. So where you find this Colonic cells, ma? So below the epidermis. Epidermis means what? Epidermis means outermost layer of the plant is called epidermis. And below the epidermis, what are the type of cells you will find? Colonic cells you will find. Are you able to understand, ma? So it is found as a homogeneous layer or in patches. What is the meaning of this statement? They are present in homogeneous layers or in patches means what? Homogeneous means all the cells of this Sclerenchyma, they may be continuous. Okay, ma? Or just you can find them in patches also. It means some group of cells are present here, then they are discontinued, some other group of cells are found somewhere. Then this kind of discontinuation is called as arrangement in patches. For homogeneous layers, they are present continuously throughout the region below the epidermis. Are you able to understand, ma? Okay. So it consists of cells which much thickened corners due to deposition of cellulose, hemicellulose and pectin. Are you able to understand? Intercellular spaces are absent. In the case of parenchyma, cells they are made up of what? Cells they were made up of cellulose. Are you able to understand, ma? And intercellular spaces were also present. Where? In parenchyma. But here in collenchyma, you don't find any intercellular spaces. What is the reason? The reason is that in the gaps between these cells, so deposition of cellulose, hemicellulose, and pectin will be present. Are you able to understand, ma? For example, here in the figure you can see here. So these are the cells, no, ma? Cells are arranged like this. Are you able to understand? So definitely in between the cells you will find intercellular spaces. No? But these intercellular spaces, they are filled by what? They are filled by some depositions of cellulose, hemicellulose, and pectin. So these materials they come and settle in between the cells of this collenchyma. That is why you don't find any gaps between the cells. Are you able to understand? ఏవి డిపాజిట్ అవుతున్నాయి చెప్పండి? Cellulose, hemicellulose, pectin. They are getting deposited. That is the reason why you don't find any gaps between this collenchyma cells. Fine. So here the structure of collenchyma cells it is given. You can see here. So thickened corners are present. Protoplasm is present. Vacuole is present. Cell wall is present. So these are the structures of collenchyma cells. Okay, ma? So they may be continuous also. They may be in patches also. Where they are present below the epidermis. Are you able to understand, ma? And you don't find any intercellular spaces. Clear? So next we are studying about Sclerenchyma. Are you able to understand, ma? So it consists of long narrow cells with thick and lignified cell walls having few or numerous pits. So on the cells of this Sclerenchyma, you will find some pits also. You can find either one pit or you can find multiple pits also on the cells. Pit means no guntan pit anta. Are you able to understand, ma? So on the cells of Sclerenchyma, you will find some pits also. Single pit may be present, numerous pits also may be present. And what they are made up of, ma? They are made up of lignified cell walls. So a material called lignin, that is the material of which these Sclerenchyma cells they are made up of. So what is the material they are made up of, ma? Lignin. So that is why they are called as lignified cell walls. Clear? So they may be long or they may be narrow, thick and lignified cell walls. So that is the construction of Sclerenchyma. So they are usually dead without protoplast. So all these cells they are dead cells. Ma. Clear? Usually dead. It means they may be alive also. Clear? But usually they are dead and you don't find any protoplast. What is meant by protoplast? Protoplasm means what? The combination of cytoplasm and nucleus. That is called as protoplasm. Are you able to understand, ma? So here in these cells, almost they are dead. You don't find any cytoplasm. You don't find any nucleus. Are you able to understand, ma? So that is why they are called as without protoplasm. Clear? So based on the form, structure, origin and development, Sclerenchyma is of two types. So depending on the structure and how they are developed, these Sclerenchyma cells, again we are dividing them into two types. What are the two types? One is a fiber type of cells and the second one is sclerids type of cells. Are you able to understand, ma? So fiber type of cells means this is the shape. So this is the type of the shape which you see. So they are formed like elongated structures. You know? And sclerids means this is the shape of the structure which you find. Sclerids. Are you able to understand? So here, let us see what are the structures which are present. So this is a fiber, clear, and this is a sclerid. So depending upon the shape and development, Sclerenchyma again we are dividing into two types. One is a fiber type of cell and other one is a sclerid type of cell. So lumen is present, thick cell walls are present. Clear, Nama? Lumen is present here also, pith is present, and a thick cell wall is present. Are you able to understand, ma? So pith, where you find pith, you will find in sclerids. Are you able to understand, ma? So inside the cells, you will find soft tissues, ma? They are called as pith. So where you find the pith, in fiber or in sclerids? In sclerids, you will find the pith. So this is another important point which you have to remember. Are you able to understand, ma? That's it. So some more information he gave here about fibers and sclerids. So these are thick walled, elongated and pointed cells. So we are discussing about this one, fibers. Clear, ma? So generally occurring in groups. So this is the nature of fiber. So what about their walls? Walls are thin or thick? Walls are thick. Are they elongated or not? They are elongated. Because they are elongated, they are making the shape of pointed cells. Pointed cells laga ఉంటాయి. Clear, ma? And generally they occur in groups. Then what about sclerids here? They are spherical, oval or cylindrical, highly thickened dead cells with very narrow cavities, which is called as lumen. So they are found in fruit walls of nuts, pulp of trees like guava, pear and sapota, seed coats of legumes and leaves of tree. So this is the presence of this second type of Sclerenchyma cells, which is called as sclerid. Are you able to understand, ma? So what is their shape, ma? They may be spherical, they may be oval, they may be cylindrical. Are you able to understand, ma? So highly thickened dead cells. It means the cell walls are very, very thick and they are dead. So where you find them? You will find them in the seed coats. Seed coats on the seed paina coating untundi kada. Shells will be present, nama. So these shells, they are made up of what? They are made up of Sclerenchyma sclerid cells. అండర్స్టూడ్, బికాస్ దే ఆర్ డెడ్ సెల్స్. ఫర్ ఎగ్జాంపుల్ ఒక బాదం పప్పు తీసుకున్నారు అనుకోండి, దానిపైన షెల్ ఉంటుంది కదా. ఈజ్ ఇట్ ఎ లివింగ్ షెల్ ఆర్ ఈజ్ ఇట్ ఎ డెడ్ షెల్? డెడ్ షెల్. సో ఆల్ దీస్ సెల్స్ ఆఫ్ డెడ్ షెల్స్, దే ఆర్ మేడ్ అప్ ఆఫ్ sclerids, which is a part of Sclerenchyma. Understood, Amma? That's it. So where you find them, you will so narrow cavities are also present. In these cells you can find narrow cavities, and these narrow cavities you can call them as lumen. Are you able to understand? Cavities ante gaps ఉంటాయి. So those gaps you can call them as lumen. Are you able to understand? Okay. So they are found in fruit walls of nuts, pulp of fruits like guava, pear and sapota. Similarly in the seed coats of legumes and leaves of tea. Tea leaves, you know, no? So in the leaves of tea also you can find this sclerid type of cells. So what is the function? Function is it provides mechanical support to the organs. So what is the function of Sclerenchyma cells, ma? These tissues, they provide support to the plant and different parts of the plant also. Clear, Nama? So then we are studying about the second type of permanent tissues, which is called as complex permanent tissues. Already we studied that permanent tissues, there are two types, Nama. One is simple and the second one is complex. In simple, how many types? Again we have three types. Are you able to understand, ma? Parenchyma, Colonyma and Sclerenchyma. And what are their functions? Already we have studied. Now we are studying about the second type of tissues, which is called as complex. So what is the difference between simple and complex? In simple tissues, you will find only one type of cells. But in complex tissues, you will find cells which do different functions. Are you able to understand? That is the difference. So therefore, there are more than one type of cells and they work together as a unit. Okay, ma? In simple tissues, you will find only one type of cells. But here you will find many types of cells are present and they work together to produce some function. Are you able to understand? So therefore, again there are how many types, ma? One is xylem and the second is called as phloem. Okay. So see the structures here, ma. In the figures you can see the structures. For example, if you take a longitudinal section of the stem, or you see a longitudinal section of the stem, then this is the shape of the cells which you can find. So they are thin-walled parenchyma cells are present here. Are you able to understand, ma? And this structure you can call them as the xylem. And connecting the xylem you can find the phloem. Okay, ma? And what is this one? This is thick walled cells which add mechanical strength to the stem, and you will find the outermost layer which is called as epidermis. Are you able to understand, ma? So these small cells which you are finding, that is called as xylem. Xylem is again connected to phloem, and then there are some middle cells which give mechanical strength to the stem, and outer covering is called as epidermis. Okay, and similarly, here we have some more diagrams now. How xylem and phloem are arranged, it is present here. So this structure you can call them as the xylem, and the next structure you can call it as the phloem. And again, if you take the magnified look of this one, then up to here you can call it as xylem, and the next structure you can call it as the phloem. Are you able to understand, ma? Clear? So this one is phloem, ma. So this is the xylem, and both of them they have a small gap between them, which is called as cambium. So already we studied there are three types of cambium, nama. So this one arrangement of cells is also called as cambium. So in between xylem and phloem, you can find another small group of cells which is called as cambium. Are you able to understand? And what is the function of xylem and phloem? We will continue. So therefore, complex permanent tissues, there are how many types, ma? So two types. They are xylem and phloem. So which is the group of complex permanent tissues which is towards the center of the stem, xylem or phloem? Xylem you will find towards the center of the stem. Okay, ma? Phloem you will find on the outer side of the stem. Are you able to understand, ma? Or towards the end of the stem. That's it. So first of all, we are studying about xylem, the structure which is a part of complex permanent cells or complex permanent tissues. So what is the functions of xylem? Let us see. It functions as a conducting tissue for water and minerals from roots to the stems and the leaves. Are you able to understand, ma? So what is the function of xylem here? What xylem will do? So xylem is responsible for transporting water also, for transporting the required minerals also. So we know that the plants they require a lot of micronutrients, no, ma? And these micronutrients they are taken by the roots and supplied to the plants. Are you able to understand? And who supplies this water and minerals, no? They are supplied by the xylem. Clear? And what is the second function of this one? It also provides mechanical strength to the parts also, to the plants also. And it is composed of four kinds of elements. So one is called the trackets, vessels, xylem fibers, and xylem parenchyma. Are you able to understand, ma? So xylem is again divided into how many types, ma? It is divided into four types of elements. One is trackets, vessels, xylem fibers, and xylem parenchyma. Clear? So in the figures you can see here, ma. So these structures you can call them as trackets. So you are seeing some this type of structures, ma. They are called as trackets. And surrounding the trackets, you can find these small cells which are called as parenchyma cells. You understand? That's it. So four parts of xylem we are supposed to remember: trackets, vessels, xylem fibers, and parenchyma. We will study each of them. Okay. So first of all, we are studying about trackets. First type of uh, part which is present in xylem. Clear, ma? So see the structure which is given here, ma. So in xylem, you can find this xylem fibers which are present here. So these structures you can call them as xylem fibers, and this structure you can call it as xylem parenchyma. Clear, ma? Then you will have this kind of structures which is called as xylem vessels, and this total structure you can call them as trackets. Clear? So totally xylem is made up of four different type of parts. So xylem fibers, xylem parenchyma, xylem vessels, and trackets. Now let us study about trackets. Clear, ma? So these are elongated tube-like dead cells with thick and lignified walls and tapering ends. So this is the description of a trackets. So one important point is that it is a dead cell. Are you able to understand? Trackets are basically dead cells. So what is the type of walls they have? Thick walls are present and lignified cells are present. Lignified walls. What is meant by lignified walls? They are made up of a material called lignin. That is why they are called as lignified walls. And they are elongated tube-like structures. Okay, ma? So in the figure, you can see this, no, ma? So these, they are called as trackets. So they are elongated, no? Each cell is elongated. Are you able to understand, ma? They are dead and they are made up of lignin. And tapering ends. What is meant by tapering ends, ma? It means when you go to the what you call the last position or ending of this cell, then they become very narrow. So narrow structures are created, no, ma? So these narrow structures you can call them as tapering ends. Okay. So these are the properties of trackets. So they are long, they are dead, walls are thick, made up of lignin, and ends are becoming narrow, which is called as tapering ends. Clear? Protoplasm is absent. They are dead cells. No? How can you find protoplasm, cytoplasm, nucleus, different organelles? You don't find in this cells. Clear, ma? Inner layer of cell walls have thickenings which vary in form. So inner walls of tracheids, they are very thick and they vary from one trachea to one cell to another cell. That is the meaning of this one. So in flowering plants, tracheids and vessels are the main water transporting elements. So therefore in flowering plants, which are the main water transporting elements? Ma? Tracheids and vessels. Xylem is made up of four parts. No? So out of these four parts, which are responsible for water transportation? Tracheids and vessels. So this is the point which you should remember. Clear up, clear now? So remember up to here, we will continue in the next class. Clear, ma?